Please welcome Sara Beery, Assistant Professor of Electrical Engineering and Computer Science, MIT.
Hello, everyone. Like was just said, my name is Sara Beery. I'm an assistant professor here in the AI and decision-making department. But my research and my group's research really tries to think about one of what I'm sure all of us are aware is a huge ongoing challenge, which is global biodiversity loss. So we're currently in what's being deemed the 6th mass extinction of biodiversity. We are losing species at a faster rate today than we were during the last extinction of the dinosaurs. And of course, this is related to anthropogenic change, to the sort of spread of development globally. It's also related to climate change. But the thing is, there's so much we actually don't know about why this is happening, about which species it's affecting the most, and about how we need to think about stability and regeneration for biodiversity, looking to the future, looking to those next generations. So today I'm going to be talking about mechanisms that me and my researchers in my group have been developing to try to enable novel scientific discovery using multimodal AI systems. So, like I said, biodiversity is in catastrophic decline. But the important thing is we're not just losing species, we're actually losing populations. We currently have estimated by the World Wildlife Fund about 70% less wildlife alive on Earth than we did in 1970. That's a huge number. And that biodiversity, this sort of just existence of living wildlife on Earth, is intrinsically tied to a bunch of things that are of social importance as well. Things like climate change. Biodiversity actually impacts climate change too, in the other direction. Biodiversity is one of the big factors in what's called bioturbation that takes forest carbon, which is on a life cycle of about 100 years, and transmits it into soil carbon, which is a life cycle of closer to 1,000 years. So when we're thinking about carbon emissions, we really need these mechanisms, these sort of complex ecosystems that help transmit carbon into more stable storage systems. Biodiversity is also tied to public health, particularly with the spread of zoonotic disease, things like COVID. Biodiversity is tied to food security. It turns out when you have more diverse pollinators, you have more nutrition in the food we grow. So it's beyond just needing pollinators to be able to grow food. We actually need diverse pollinators to get nutrition from our food. And biodiversity is tied to ecosystem services, things like carbon capture, things like cleaning the air that we breathe. Ecological data, people are trying to understand these things, and we're collecting data now with both increased quantity and increased diversity. We, in my group alone, work with everything from satellite imagery, low-flying aircraft and drones, on animal sensors. These are things like collars placed on elephants or seals or anything you can imagine to actually track the animals, but also collect data that we don't have, things like ocean temperature, ocean salinity, in ways that are actually intelligently collected. Huge networks of stationary sensors, things like camera traps, bioacoustic sensors, collecting information about species on the ground all over the world. And then increasingly what we call community science data. So this is, I don't know how many in this room have tried to use iNaturalist or are familiar with Merlin Bird ID. Awesome. If you haven't, I think these are really amazing platforms. They let passionate community members go out and collect scientific data of the species they see that then get identified by the community to research grade and get transmitted into the Global Biodiversity Information Facility as species occurrence records. And the funny thing about biodiversity, all of these different types of data, is it turns out none of them is giving us a direct measurement of what we care about. None of them is giving us a species or a direct sort of estimate of what we call alpha diversity, the number of unique species in a given place. They're all proxy data. They're things like pixels in images or sonar signals. And all of this needs to be then processed into scientific insight, something we can actually make a decision on. And that's where we need AI. And AI is currently used to process data for thousands of conservation organizations, thousands of scientists worldwide. We do things like detect humans, animals, and vehicles in static networks of cameras, to enable state agencies, governmental agencies, to get to the point where they have good, actionable, up-to-date information to make policy decisions about, for example, wolf populations in the Idaho. But it turns out that that environmental data across all of those different data types is really not collected uniformly. We have this estimate on the left of alpha diversity worldwide, which is clearly mostly in the subequatorial tropics. If you look at where we have that species occurrence data in the Global Biodiversity Information Facility, it's almost anti-correlated. So there's clearly a huge need to go out and get information, to have these mechanisms for extracting information or collecting information in places where we maybe need it most in order to maintain that sustainability and the regeneration of biodiversity. And so looking at all of these different data types, for the rest of this talk, I'm going to focus just on iNaturalist data, just on one of these types of data, and bring up what I think is an exciting direction that helps us get more information out of the data we've already been collecting and helps us think about mechanisms to scale that data collection. So these are all images from iNaturalist. And iNaturalist has now collected over 173 million observations of species, over 460,000 species. This is actually now 95% of all of the biodiversity data that we have collected throughout history. So just enabling passionate community members to go out and be a part of this system, to be a part of understanding biodiversity, is clearly incredibly valuable. But currently, what we have in iNaturalist is an image with a species label. So for example, in this image, the iNaturalist label is plains zebra. But there is way more scientifically useful information in this image than just the presence of a single species. We can see behavior, potentially some sort of conflict behavior. We're seeing wildlife and livestock coexistence. There's a lot more here that we would love to be able to get to than what is identified by the community. And this is kind of ongoing. So for example, you could find examples of hermit crabs using plastic waste as its shells, these intersections between human waste and wildlife. Things like wildlife using man-made roads, intersections between wildlife and human infrastructure. Or even stuff like all of these other queryable systems that would let us get more information from the data that we've already collected. So the hope here is that we could actually build systems that query using multimodal AI to enable us to get at all of that diversity information in those 170 million images. So what we're trying out is we're using multimodal AI models, vision language models like CLIP, to extract embeddings. These are low-dimensional representations of these images that are semantically meaningful. And then what we can do is we can actually also extract an embedding from any text query, such as a California condor tagged with a green 26, which would enable us to track an individual condor in those sightings over time. Then what we can do is we can just find a similarity score across these representations of both images and that text query, and then we can re-rank all of those images, all 170 million images, to find the ones that are closest to that interesting text query. And then what this gives us is a mechanism to make it easy for an ecologist, for a scientist, for a community member, for a local area manager to get to useful information to ask new questions from the data we have. So this is great, but it turns out that these existing models are insufficient to do this for scientific data as is. Turns out there are a lot of different types of low precision errors. So here it's doing a good job of finding green tags on a condor, but they're not the correct number. Or for scientifically nuanced information like allo-feeding behavior, you're not getting relevant examples. So what we're looking at now is developing re-ranking mechanisms using now more intense, more computationally expensive vision language paired models to try to get better results towards the top of that retrieval system. And so now what this looks like is you re-rank maybe the 2,000 closest matches from CLIP using something like an LVLM, and we get much more accurate results. And so here, what you can see is that this re-ranking, it makes this even better. It makes this work better for scientists. And LVLM is expensive, right? If we wanted to run a single query using LVLM over all 170 million images, it would take about 57 days. But by kind of pairing the efficiency of a standard clip model with the accuracy of these LVLM models, we can find a sweet spot where you can get a return from 170 million images that's accurate in about 2 minutes. And of course, we're working on trying to make this more efficient. And what we've been doing now is we're working directly with ecologists to try to design these interactive multimodal systems that make this process really easy for them to use. So we've deployed this with the team at iNaturalist. Pretty soon it's going to be going out actually for public use, which I'm very excited about. But we're also working with ecologists across many different areas, Ocean Vision AI, for example, to try to translate these into operational systems that are letting us ask and answer these new open-ended questions from data that we've already collected, which I think is very exciting. So thank you very much. If you have any questions, or if you're interested, or if you're working with biodiversity data and you'd like to think about how AI could play a role, please reach out. And I'd also like to thank the other researchers who helped work on this project.